We are going to demonstrate the gram stain technique. We utilize a gram stain in the microbiology lab to help with microscopic identification of bacterial organisms. The materials needed are a sterile slide, a QC slide which is incorporated with a gram-positive and a gram-negative organism. We need our bacterial isolate, sterile inoculating loops, and our gram stain reagents, which consist of methanol to fix the slide, crystal violet as the stain, iodine as the mordant, alcohol as a decolorizer, and safranin for the counter stain. Before we begin, we will label our slide with a name, the date, and initials. If this were a patient specimen, you would also record the source of the specimen on the slide. We must utilize the quality control slide in order to assess the method and the technique that was used in the gram stain procedure. We will first pick an isolated colony, inoculate the slide, let that dry for a minute, and we will add the fixative of the methanol, and we will let that methanol air dry. You do not want to start the gram stain procedure until the methanol has completely dried on the slide. All right, next we will stain our slide with the crystal violet. And we will allow that crystal violet to sit for 30 seconds. After the 30 seconds, we will rinse with water, and we will tap off the excess water. We will now add the iodine, and the iodine will sit for 60 seconds. So when we added the crystal violet, the cell wall will take in that crystal violet color, and we add the iodine as the mordant to set that color into the cell wall. After the 60 seconds, we are rinsing with water, and again we'll tap the excess water. And our next step is a decolorizer. So if the organism was not able to take in the crystal violet, when we rinse with decolorizer, the crystal violet will wash out of the cell wall. We will do the crystal violet for approximately 10 seconds, or until all the crystal violet has run off. We then add safranin, and we allow the safranin to sit for 30 seconds. And the safranin acts as a counter stain. So for the organisms that are not able to maintain the crystal violet stain within the cell wall, when we add the crystal violet, that red will be absorbed into that cell wall, and we will see that under the microscope. So after 30 seconds, we rinse. and they can air dry, or you can blot dry with a paper towel or Kim wipe. And to blot dry, you just fold it over, blot it. Do not rub it. If you rub it, you will rub the organism off. And the gram stain is complete. So we then go to the microscope and we look at the microscopic morphology. And you can see that we have purple and red, pink organisms. The purple organisms, the round, are called gram-positive. The pink to red are gram-negative organisms. So our gram-positive organisms are able to hold on to that crystal violet, and you can see that the E. coli or the gram-negative was not able to hold on to the crystal violet after the decolorizer was added, and when we added the counter stain, it turned it a pink to a red. We also, when we view microscopic morphology, we are looking at the shapes of the organisms. So we can see that our gram-positive organisms are round, and the round are called cocci. Our gram-negative organisms are rod-formed or bacilli, and we refer to those as gram-negative bacilli. Another form you might see is a coccobacilli, which takes on the characteristics of both cocci and rods. Occasionally, you will see gram-variable organisms, where the organism actually retains some of the crystal violet, but not all of it, and therefore stains purple and red.